Hi there, my name is Ruth Wilson, and today I'm going to be talking to you about transgenic animals. So, what are transgenic animals? Transgenic animals are animals that have had a foreign gene deliberately inserted into their genome. In other words, their DNA has been manipulated through various molecular techniques to achieve a desired goal. This involves the insertion of a foreign DNA into the embryo of an organism before birth. Examples are glowing rabbits used to study diseases and the way that it moves within the body, transgenic pigs that serve as important biological models for human diseases, as well as enviropigs that produce more environmentally friendly waste, featherless chickens for food sources in warmer climate regions, as well as transgenic cows that produce more milk in a shorter amount of time. Now let's talk about the first transgenic animals. Transgenic animal technology is a relatively recent innovation in the genetic engineering field, dating back to 1973 when Stanley N. Cohen and Herbert Boyer developed recombinant DNA technology. They succeeded in altering the genetic makeup of E. coli by introducing a foreign gene from another type of bacteria. In 1974, Rudolf Jaenisch, a professor of biology at MIT, created the world's first transgenic mice by inserting viral DNA into their genome, which contained the SV40 leukemia genes, which were also transferred to their offspring. Then, in 1982, Ralph Brinstern of the University of Pennsylvania carried out research on our DNA by inserting the structural genes of human growth hormone into the embryos of mice. He then noticed a significant difference in the size of the mouse with the foreign gene. The transgenic mouse grew larger in size compared to its counterparts. So how are these animals created? Since the inception of transgenic animal technology in the 1970s, scientists have made commendable steps in enhancing the efficiency of the procedure. While there are a variety of approaches in creating transgenic animals, I will be talking about the most common methods: DNA microinjection, embryonic stem cell mediated gene transfer, and retrovirus mediated gene transfer. DNA microinjection seeks to expose a fertilized egg to the transgene before the onset of the cell differentiation process. This is done in vitro by using a microtube suction device to hold a newly fertilized egg and a separate ultrafine glass needle to insert transgenic DNA into the male pronucleus. Upon successful procedure, the resultant organism will have all its cells and tissues containing the essential gene inserted during the process. The egg and sperm used for this experiment undergo fertilization in vitro before the fusion of the two pronuclei inside the zygote, as well as the microinjection of the male pronucleus with the rDNA. The manipulated fertilized ovum will then be inserted into the host female's oviduct. This foster mother would have been induced with a castrated male prior to insertion. Although DNA microinjection technology has been efficient and prominent in the past, it has had a fair share of its challenges, like variable expression patterns and random integration. In other words, the process is very random, and the gene may underexpress, overexpress, or not express at all. Unlike DNA microinjection, which makes the male pronucleus transgenic, embryonic stem cell manipulation uses the embryonic stem cells. After undergoing in vitro fertilization, the embryo passes through a culturing process to the blastocyst stage. The desired stem cells are then harvested. The desired transgenic gene is inserted into the inner cellular mass of a new blastocyst, followed by the introduction of the manipulated embryo into the uterus of a pseudo-pregnant host. This technique works really well in mice. The use in larger animals is still being explored. Retroviruses involve organisms which carry their genetic materials in RNA form rather than DNA. The retrovirus-aided technology is essentially the transfer of a gene to the host cell using a retrovirus as a vector. The genetic material is taken into the cell by infecting the host cells, which is the virus' natural method of reproducing. This process results in chimera, which is an organism consisting of cells from different zygotes in which not all cells carry the retrovirus. Chimeras are then inbred for 10 to 20 generations until homozygous transgenic animals are obtained and the transgene is present in every cell. The downside to this method is that the live birth of animals containing the transgene is extremely low, and a lot of times this manipulation can lead to a disruption of the animal's genome, causing abortions or adverse health effects. Transgenic animals have become important to humans in three primary sectors: agriculture, medicine, and industry. In agriculture, farmers have utilized this technology to breed specific animals with desirable traits such as increased milk or meat production. It has also enabled farmers to harvest high yields in a short time, thereby addressing food challenges currently witnessed in many parts of the developing world. Also, they are able to create animals that are more resistant to disease by manipulating their genes. In medicine, there has been development of transgenic animal models that are able to display symptoms similar to diseases exhibited by human beings. That way they can be studied. As far as industry goes, we are able to make products such as antibodies, insulin, growth hormone, and anti-clotting factors that come from transgenic rabbits, cows, goats, and pigs. Also, enhance safety of certain chemicals by producing toxicity-sensitive transgenic animals has been extremely useful. Another example is transgenic pigs with the capability of digesting plant phosphorus more efficiently than conventional pigs. We have talked about the three major sectors where transgenic animals are being used, and now here's a short video that shows the ongoing research and the people involved in making these transgenic animals.
From anti-aging therapy in mice, animals carrying human cells, breeding the healthiest livestock, and building organs outside the body, genetic engineers are constantly searching for answers to many social challenges. From global issues to medicine, these guys put everything to the test. One of the biggest goals of genetic modification is to reduce world hunger by producing more food. These featherless chickens have been genetically engineered to keep them cool.
Since I'm a geneticist, I was looking for a genetic solution. That means instead of cooling the environment of chickens with feathers, is helping them feel cool by just removing the feathers.
Without feathers and scales on their feet, these birds can grow comfortably in hot countries like Nigeria and Indonesia. The hope is that once successfully bred, the healthy commercial-sized birds will help feed underdeveloped countries. To fight global warming, some turn to enviro pigs. The industrial amount of waste from pigs is a serious environmental pollutant.
Pig manure has a very high level of phosphorus within it from normal diets through normal pigs. And that phosphorus usually is more than is required by crops produced on land fertilized with pig manure.
But the remarkable thing is that the enviro pigs carry a man-made gene. One part comes from E. coli bacteria, which produces an enzyme that can break down phosphorus. And the other from mice produces a genetic switch that allows this special enzyme to be made in saliva. These two pieces of DNA were combined to form a new compound gene that makes their poo more environmentally friendly. Medical researchers across the world are also turning toward transgenics for answers.
If you look at the word transgene, this means that the gene was transferred, a foreign gene was transferred to the animal.
Having successfully used transgenics to insert a jellyfish gene into a rabbit, when charged with blue ultraviolet light, the skin and the eyes of these rabbits glow green. But why make animals that glow in the dark? The incandescent gene allows researchers to easily track the movement of cells. By tagging certain cells with the glowing jellyfish genes, scientists can track which cells go where after an organ transplant. The technology is helping to find treatment for problems like blindness and bone disease. Glow-in-the-dark rabbits are just a start to the medical challenge that genetic engineers have taken on. No one said it was easy being green.
Overall, the technology presents enormous benefits to our society, including increasing food production, the use of transgenic animal models to study and cure human diseases, and a novel way of livestock breeding. The creation of transgenic animals is resulting in a shift from the use of higher order species to lower order species, and is also affecting the numbers of animals used. Transgenic animals have the potential to improve human welfare, as it has made it possible to consider employing cattle, swine, sheep, and goats as processing units to manufacture proteins or as organ donors. Transgenic animal technology plays a critical role in meeting the ever-increasing demands of the global population, particularly by increasing the efficiency and the success of livestock breeding and production, as well as minimizing environmental impacts.